Recep Adiyaman, and a postdoc at the University of Reading, working with Professor Liam McGuffin. Today, I will talk about the improvement of protein tertiary and quaternary structure prediction using the ReFOLD and multifold refine. Here is the outline of my presentation. I will talk about the refinement of AlphaFold2 models, refinement methods, performance of ReFOLD and multifold refine, and the server applications. Here, we questioned can we refine the AlphaFold2 models further. The refinement aims at increasing the accuracy of predicted 3D structures, as the accuracy is crucial for further computation studies, such as drug discovery. AlphaFold2 models are highly accurate near experimental accuracy, so the refinement category was dropped from CASP. However, there are still rooms for improvement, and error can be fixed by the refinement tools. And here, we try to answer whether we can refine the AlphaFold models or not. Here, we categorize the refinement tools in two main categories. Molecular dynamic methods has been very popular during the CASP experiment. Neural network-based methods has been promising since the AlphaFold2 developed. In this research, we aim to combine the molecular dynamic-based and neural network-based refinement methods. As you can see, the refinement methods has some pros and cons. May capture the atomic level dynamics, rationalize the protein interactions, but could be computationally expensive and limited improvement could be seen in the predictions. Neural network-based refinement has been trained on the experiment data, and effective refinement could be provided and could be much faster than and less computationally intensive than the molecular dynamic method, but they may have limited capture of real protein structure and has some issues to stoichiometric aspects due to the experimental data. We have been pioneering the usage of the local quality estimation to guide the molecular dynamic-based protocols to refine the protein structure since CASP 13. Here, we adopted our ReFOLD pipeline to refine the AlphaFold models using the fine-grained restraint strategy based on the PLT score generated by AlphaFold2. Simply, we use the PLT score as the first constant to restrain the related residues. In other words, stronger restraint was applied to the well-predicted residues and less strong restraint was applied to the less accurate regions based on the PLT during the molecular dynamics simulation. We have also used very modest computational resources compared to other CASP models. You can even simply run on a standard desktop. We also aim to provide the conformation landscapes of AlphaFold2 models while maintaining the high accuracy due to the restraint strategy. When we look at the performance of ReFOLD for the CASP 14 targets, ReFOLD managed to prevent the 3D models from structural deviation, while there is a modest improvement according to GTTS and LDT score, there is a significant improvement in more probability score. So we can say that ReFOLD managed to improve the stoichiometric quality of the structures significantly. As mentioned, AlphaFold neural network has been very valuable for the community, as it has been very well trained on experimental data and lead to high accuracy. The community was able to use the neural network for further improvement with custom MSA, custom templates, and more statistical options. The development of CollabFold also made possible to run AlphaFold2 with less computational resources. We used the custom template option to feed the AlphaFold2 neural network with best prediction from top 5 groups in CASP 14. We also used more statistical option to refine the predictions further through the AlphaFold2 neural network. We call this protocol as multifold refine, implemented in our server and manual prediction pipelines in CASP 15. We also refined CASP 14 single-chain targets using the multifold refine method. The predictions were significantly improved according to LDT and TM score, while ReFOLD showed significant improvement according to more probability score. As in this figure, multifold refine managed to provide better prediction than the Bayesian methods according to LDT and TM score. Bayesian methods, I mean the templates we use to feed the AlphaFold2 neural network. AlphaFold2 multimere has enabled to predict the quaternary structures, so we used the AlphaFold2 neural network to refine the quaternary structures to improve the accuracy beyond the AlphaFold2 predictions by feeding custom templates. The accuracy of the predictions are significantly improved according to LDT, TM, and QS scores. As you can see that the majority of the 3D models generated by multifold refine are significantly better than the baseline models according to TM score. It also performed well according to LDZ scores. We also used the multi-fault refine in our CAS15 pipeline. We were the top performing group from the UK and ranked 6th in the regular prediction, 11th in the multi-target predictions. Multi-fault refine method was also implemented in our server pipelines and ranked 9th on monomeric, 8th on the multimeric target prediction categories. It's also worth to mention that multi-fault significantly outperformed the based on standard ALPHO2 and ALPHO multimer as well. Our methods are also presented as web servers. These are freely available. We also continue to develop the info servers more than a decade. When you submit your protein sequence, we provide different protein conformations, quality estimation, protein ligand interaction, and disorder region prediction for your sequence. After the submission of your sequence, you receive results within 48 hours. When you receive the predicted 3D models, you can have option to refine the prediction further using our ReFOLD 3 web server. You can also separately submit your 3D model to be refined by our ReFOLD server. You should receive the results within 48 hours again. Multi-fault server was also designed to predict quaternary structures. You can also provide stoichiometry of the structure. If you don't know, we will use template to build the quaternary structures. This is an example output of multi-fault server web page. 5 models will be provided and ranked by the PLDD scores. Multi-fault and multi-fault refine methods Docker image is also available. You can download Docker image and then run your own local machines as well. In conclusion, ReFOLD is successful in preventing the molecular dynamic simulations from structural deviation and a risk-averse method for providing the confirmation landscape of ALPHO2 accuracy level for further computation studies. We have demonstrated that ALPHO2 algorithm can be used to refine the 3D models. Multi-fault refine is advantageous due to the low computation resources required. 3D recycle also seems to be optimal for monomers, but 12 for multimers. The combination of ReFOLD and the multi-fault refine would be more beneficial for further computation studies, such as drug discovery. We have also published this research in Bioinformatics Advanced. You can check out the paper for further information. Thank you for listening. We also thank BBSRC for funding our research. There are also our references, you can read more about them.